I have the pleasure today to be moderating the session and introducing our wonderful speaker for the day. So just to give a bit of background about our speaker, today we have Professor Adesola Ogunniyi, and he's a clinician, researcher, neuroepidemiologist, and professor of medicine at the College of Medicine, University of Ibadan. He's a consultant neurologist at the University College Hospital in Ibadan, and was the director of research and later site principal investigator of the Indianapolis-Ibadan Dementia Study that lasted 20 years. Thereafter, he became the principal investigator of the identification and intervention for dementia in early Africans, the IDEA study, which was funded by the Grant Challenges Canada. His research interest includes the cross-cultural study of dementia, the epidemiology of other neurodegenerative diseases, epilepsy, and stroke. And so today, I think he's going to tell us a bit more about the IDEA study, as well as the interplay between epigenetics and dementia. So with that, I'm going to hand over to Professor Adesola. And just one thing, if you have any questions, please write them in the Q&A tab, and then we'll get to them after the talk. So thank you so much, Professor, for being with us, and we're looking forward to this talk.
Thank you, Natasha, and I'd like to welcome everybody to this webinar. I'd like to start by thanking International Parkinson's Disease Genomics Consortium for giving me this opportunity to use the platform to talk about dementia. They are related, but at least I'm not doing a lot on genomics at the moment, just talking about environmental factors that work on DNA and result in clinical presentation of dementia. I'd like to thank the organizers for allowing people like Professor John Hardy and Professor Zetterberg to go before me, and I really enjoyed their sessions. I hope this will be interesting because I'm supposed to inspire up-and-coming researchers on what to do in terms of their career. The agenda is to talk a bit about my background and career journey. Then I'll talk about the challenge of dementia. I'll then go on to biomarkers and epigenetics, talk a bit about hypertension as a risk factor for dementia and the risk marker for dementia. Then I'll then close and then do some acknowledgement. Now, let me apologize at the onset that I was limited to 5 to 6 slides for my research presentation. So a few of the slides may appear crowded because the essence is to allow me to talk to the slides so that people can understand what I've gone through. So I'll begin with my career journey. And on this slide, I have various institutions that have been presented. My undergraduate medical education was at Obafemi Awolowo University, Ile-Ife. I was among the first set of medical students there. I entered 1971 and 19th degree in physiology, which I'll talk about later, what that one did to my career. Eventually qualified as a doctor in 1978 and started residency training at University College Hospital, Ibadan in 1980. And I completed my fellowship training in neurology in 1985. Thereafter, I proceeded to the National Institutes of Health, USA, with the Fogarty International Fellowship, and I had opportunity of training in neuroepidemiology. At the NIH, I had the fortune of undergoing courses at the NIH Graduate School in epidemiology and biostatistics, which I found very essential for research as a neuroepidemiologist. And eventually returned in 1987 to pick up a job again as senior registrar in UCH between May and October 1987. My academic career began with joint appointment in physiology and medicine in 1987, and I eventually moved on to medicine in 1988, and I've remained in medicine since then. My research focus has been dementia, and in 1991, I won the International Neuroepidemiology Award that was started by the American Academy of Neurology in memory of Bruce Schomburg, who was the chief of the neuroepidemiology branch of NIH, where I did my fellowship. This came about when we did the first community-based study of research on dementia in the DECON community, and we showed that there was relative rarity of dementia in that community. We utilized a new instrument that we just developed called the Yoruba version of the MMSC. So that's a bit about my background. Let me now go on to my research findings and research activities. And the focus of my research in dementia has been cross-cultural studies to enable me to tease out environmental factors that determine genetic predisposition to disease. And around 1984, when this started, late Professor Shutokun at that time made an impression that dementia was relatively rare in Nigerians, and particularly as Alzheimer's disease was rare. And he opined at that time that it was probably a result of some exposure in the environment that individuals in Africa did not have. And that raised a lot of curiosity. So there was that need to then show whether the substrate for dementia, which is presence of amyloid in the brain, could be demonstrated. And so our first major investigation was to look at brains of non-demented Nigerians to see whether amyloid was present in the brain. So this study that I've put there, I was written as a letter to the editor in Lancet in 1994, compared amyloid in the brain of Australians and Nigerians. And what we found at that time using immunocytochemistry was that below the age of 50 years, there was no amyloid either in the brain of the Nigerian nor the Australian. In this graph, the Australian bar chart is in orange, while the dark one is for Nigeria. But after the 5th decade, there are about the same proportion of individuals had amyloid in the brain. In the 6th decade of life, amount of amyloid in the brain of Australians approached 60%, but it was only a little over 20% in Nigerian. And in the 7th decade, almost all the Australian brains had amyloid, whereas only 40% of Nigerian individuals had amyloid in their brain. So overall, 50%, a little over 50% of the Australian brains had amyloid, whereas only 25% of Nigerians had amyloid. They also did semi-quantitative analysis and just to look at severity of amyloid deposition. It was obvious that 11% of the Australians had severe amyloid deposit, whereas none of the Nigerians had amyloid, severe amyloid deposition in their brain. The conclusion from that study was that although Alzheimer's disease appeared to be rare, the substrate was present, and so we should look out for individuals who are likely to develop dementia, and Alzheimer's disease in particular. Then, following that, the World Health Organization became interested in the problem of dementia and then invited experts from 6 countries, and they paired them. They invited experts from Canada, from Malta, from Spain, and from Chile, and then North America, USA, and Ibadan, Nigeria. With the belief that people with similar linguistic inheritance should have some genetic similarity, and so you can then tease out whatever environmental factors might contribute to dementia. Now, in case of the Indianapolis, the African Americans, as shown in this graph, majority of them were taken from Africa into the Western world many decades ago. And so comparing them with Nigerians could then result in teasing out what factors might contribute to disease. And that's what led to the Indianapolis-Ibadan study. The funding started in 1991, and the line graph shows the timeline of the study. We started recruitment in 1992 and recruited over 2,000 individuals in each site, Indianapolis and Ibadan. And we reported prevalence in 1995 and continued following them up. In 2001, because there was a reduction in number because of attrition due to death, we needed to do cohort enrichment. And so in Indianapolis, we added about 1,900 individuals, and in Ibadan, we had a little about 1,900 individuals. And also in 2001, we obtained blood from these individuals that we'll talk about when we're looking for biomarkers. And for each of these waves, we did a 2-phase study in which we did an initial screening using the community screening instrument for dementia, and participants are then categorized based on performance on the CSID into poor performance, intermediate performance, or good performance. Those with intermediate performance, 50% were invited, all the individuals with poor performance, and 5% of those with normal performance were invited to second stage review, which comprised of neuropsychological test battery, clinical assessment, and further investigations. Overall, we studied over 4,000 African Americans and almost 4,500 Yoruba, and there was 7 waves of the study over a 19-year period, and I'll present the results. Now, the first result was the prevalence data, and the prevalence data show that prevalence of dementia was much higher in the African Americans than in the Yoruba, and Alzheimer's disease was equally much higher, significantly higher, 6% versus 1.4%. And a few years later, we reported the incidence data. The incidence of dementia was about 3.2 in the African Americans, was 1.4 in the Yoruba, and Alzheimer's disease, the same thing. And these were published in 1995 and 2001. Now, you'll recall that prevalence is the product of incidence and duration of disease. So we felt that we should look at mortality, that might provide some clue as to why we may have lower prevalence in the Yoruba. And so we did a prevalence, and we did a mortality study. We looked back on our data over a 5-year period to compare mortality of African Americans and the Yoruba, and dividing them into those with dementia and those without. And what we found was that after 6 months of the follow-up, individuals with dementia started dropping off, they were dying off. But the Nigerians, the Yorubas that were normal without dementia, and the African Americans, there was no difference in the survival curve. For those with dementia, after about 21 months, the Yoruba elderly people with dementia appear to die at a higher rate than the African Americans, but over a 5-year period, there was really no significant difference in mortality. So we didn't think that mortality accounted for these differences that we observed. So we needed to look for other reasons. And the paper, for those who may be interested, was published by Parkins and Co. in 2002. So we then felt that we should then look into pathogenesis of dementia, particularly Alzheimer's disease. And the theoretical bit to start with is that the clinical expression of dementia depends on genetic factors, age-related decline, environmental risk factors, and other illnesses and comorbidity. And we had this in mind, and so we felt we should explore the environmental factors and see how this one affected genes. At this time, when we started this study, the amyloid hypothesis had already become well established, and people like Denis Selko, St. George Hyslop of Canada, Professor John Hardy were those who worked on this amyloid hypothesis and proposed it. And what it shows is that abnormal processing of A-beta at the membrane level resulted in production of insoluble fibrils, beta, that were then deposited as amyloids in the brain, in the plaques and in tangles. And this process, involving various enzymes, could be due to oxidative problems, excitotoxicity, or inflammation. And some other times could be tau hyperphosphorylation. And this resulted in cognitive and behavioral impairment, and then the syndrome of dementia. So we then decided to look at cardiovascular biomarkers. I remember that I mentioned that we obtained blood in 2001. So we explored 5 mechanisms at that time. We explored endothelial dysfunction, oxidative stress, insulin resistance, atherogenesis, and methylation, DNA synthesis. And we carried out the following tests: E-selectin, plasminogen activator inhibitor 1, homocysteine, C-reactive protein, 8-isoprostane, cholesterol, and all the lipid profile, folate, vitamin B12. We also measured insulin resistance and determined the frequencies of metabolic syndrome in our 2 cohorts. And these are the results for African Americans and the Yoruba. Now, looking at endothelial dysfunction, we found that E-selectin was increased in the African Americans, whereas PAI was higher in the Yoruba and significantly so than in African Americans. So this seemed to have cancelled out. I'll come back to talk about homocysteine, which appear to be the same at the 2 sites. In terms of C-reactive protein and isoprostane, again, we found this conflicting pattern. Higher values of CRP in the African Americans, but much higher and significant value of isoprostane in the Yoruba, which was probably adduced to their consumption of yams. Insulin resistance, metabolic syndrome, and HOMA were much higher in the African Americans than in the Yoruba. Now, can explain that in a while. With regards to lipids, all the lipid values were much higher in the African Americans than in the Yoruba. And it was important to point out at this time that about 22% of the African Americans were on statins. So the level could even have been higher still. In terms of methylation, folate was high in the African Americans, whereas B12 was one that was higher in the Yoruba. So again, they canceled out. So there were three areas that we're interested in. First one was to look at homocysteine, which appeared to be equal. So it was either there was something that was promoting the effect of homocysteine in the African Americans, resulting in cognitive decline, or the other was something repressing it in the Yoruba so that there was no expression of dementia in the Yoruba. And we did do metabolomic studies. So I'm not going to present that. And we also related homocysteine level to cognitive performance and dementia. And we found that cognitive decline was associated with homocysteine level, but it did not increase the risk of dementia. With regards to lipids, we had talked about statin. We then decided to do dietary analysis. And this was done based using dietary recall, 25 dietary recall, and actual measurement of the meals consumed and doing calorimetry to determine the constituents. And what we observed was that the carbohydrate content of the Yoruba meal was very high. It was low in fat. The protein was about 12%. Whereas the African Americans, they consumed less carbohydrate, but a lot of fat. And this will explain why they had high cholesterol level. And this was then something that was significant. At this time again, cholesterol was gaining international importance in amyloidogenesis because of the link with apolipoprotein E. Apolipoprotein E was first described by Alan D. Roses and colleagues around 1993 and was shown to be a most important risk factor for sporadic Alzheimer's disease, late onset. And so we initially did the filter cards for DNA analysis for apolipoprotein E. And so if we then look at apolipoprotein E and dementia risk. On this plot, we looked at genotype, either E4 allele or no E4 allele, and compare that as cholesterol level rose in the African American cohort and in the Yoruba cohort. The African Americans in the upper panels, the two line graphs, whereas the Yorubas and the lower panel. And you could see that in the African Americans, the risk for dementia was much higher than in the Yoruba. Those without the E4 allele, as the cholesterol level rose, the risk of dementia increased. And there was an intersection when the level was between 220 and about 250. The same pattern was observed in Yoruba, even though the risk was much lower. So we could then say that apolipoprotein E interacted with cholesterol, and that determined dementia risk. At this point, these papers were published by Morel, Kathleen Hall and Co. and Gureji. When we did the filter cards, there were only 29 Alzheimer's disease patients in the Yoruba, and we compared this with 1046 normal individuals. Subsequent study based on the paper published by Gureji, and after following up patients with dementia for about 5.2 years, and we had accrued 123 cases, we still found no association. But we then continued with prolonged follow-up, much longer follow-up for about close to 9 years. Then we had added more Alzheimer's disease cases and also diagnosed more individuals that were normal. And at this point, it was possible to then divide the individuals according to the amount of apolipoprotein E4 allele that they had, either homozygous or heterozygous. And we show that in the Yoruba, those with the homozygous had significantly increased risk of developing Alzheimer's disease, whereas those with the single copy, even though the risk was high, it was not significant. Compared with African Americans, the hazard ratios were much higher and were significant. This graph I obtained from Hugh Hendry, who compared the data in African Americans Yoruba with the meta-analysis that was carried out by Lindsay Farah in 1997. And that one showed that the risk for developing Alzheimer's disease was much higher in Caucasians than either the African Americans or the Yoruba. And if you look at the Kaplan-Meier curve, which I have there, starting from time 0, by about year 5, those with the double copy homozygous E4 allele separated from the others with single copy and those that did not have E4 allele at all and was significant. Whereas the African Americans, again, after about 5 years, you could see the separation. Both those with double copy and those with a single copy, but should add increased association with development of Alzheimer's disease. And this was a publication by Hugh Hendry in 2014. So having established that apolipoprotein E has now become a risk factor for Alzheimer's disease, we then thought we should look for other risk factors. And the one that was interesting was hypertension. Again, we had started measuring high blood pressure, we had started taking blood pressure measurements from 1997. So we had ample time to be able to follow up individuals, and we made the diagnosis in 2007. And what we found was that hypertension, both systolic and diastolic, 10 milliliter increase in systolic, 10 milliliter increase in diastolic, was associated with increased risk of Alzheimer's disease, as well as pulse pressure, which is an indication of atherosclerosis. So hypertension emerged as an important risk factor for dementia. One of the problems facing researchers or people managing dementia in developing countries is inability to be able to predict who will have the disease because we don't have facilities for assessing biomarkers. But we then felt we could use anthropometric measurements to see if we could predict those who will develop Alzheimer's disease and dementia. And so we looked at weight change over time, and we were fortunate that we were able to do this over a 10-year period. So from 10 years to the time of diagnosis, 1997 to 2007, we looked at anthropometric measurements and matched this according to the diagnosis. And we showed that the female had higher BMIs, those are the ones in the higher panel, and those in the lower in blue are the male. Now the straight line here, continuous line, and the dotted line above there represented those that remained normal. And what was obvious was that individuals that had no dementia had their weight stabilized throughout the 10-year period of follow-up, whereas those that had dementia or cognitive impairment, there was a tendency for the weight to drop. And we calculated the rate of change in weight. For those with dementia, the rate of fall was 0.19 kilogram per meter squared per year. And for those with cognitive impairment, it was 0.16 per kilogram per meter squared per year. We had an impressive number of individuals that were used for this study. We had over 1,100 normal individuals, 255 individuals with cognitive impairment, and 136 individuals with dementia. So putting all this together, we can then look at our disease model was that we had genes, we had environmental factors, we had genes interacting with genes, and we had genes interacting with environment. And we can put some of the results that are presented in this graph. So the gene that was interesting to that we're interested in was apolipoprotein E4 allele, and we showed that there was an association with Alzheimer's disease. The other genes, we're going to talk about that very briefly. For vascular factors, hypertension was shown to be a risk factor. We felt diet was also had a part to play in it. Although I don't have data for these at the moment, but we also show that individuals that were isolated had more cognitive decline and were at risk of developing dementia than those that had a lot of social activity. This was derived using the EPESE model, in which you quantified amount of involvement of these individuals, community work, religious activities, attending social gatherings, taking responsibility at home, and being engaged. So genes interacting with genes, this was an area that we wanted to explore further. And lastly, genes interacting with the environment, and we have shown that those individuals without the E4 allele, as their cholesterol rose, they were at increased risk of developing dementia. So the last bit of the puzzle was to then look at genes, other genes that may be involved. And fortunately, we were able to do GWAS study, and we were able to then compare African Americans and the Yorubas. So we had various SNPs that we studied. In the African Americans, the closest genes that were significant associated with development of dementia, Alzheimer's disease, were the ATP binding cassette subfamily 7 SNPs. And you could see here that the hazard ratios 1.84 in the African American and 2.06 for the 2 SNPs of interest, and they are significant. But neither of these was found in the Yoruba, which was quite curious. Whereas for the Yoruba, the clustering gene, as well as the solute carrier family 24 genes, were not significant in the African Americans, but were significant in the Yoruba. And the pattern was same for triglyceride, even with higher hazard ratios, which meant that all along we had resumed that the African Americans and Yoruba had the same type of genes that could predispose to Alzheimer's disease. But this latest study shows that there are other genes involved and there are differences in the genes that may be involved or predisposition to Alzheimer's disease. So I think we should then round up by presenting what are the lessons from the Indianapolis-Ibadan study, which is the essence of this presentation. And the first one is responsible conduct of research. We maintained a 20-year history of no dispute with our results, and we remain credible throughout, and the results have stood the test of time. So it's important for those who are embarking on studies like this to ensure that research is well conducted and there is good accountability of everything that goes on into the research. Second one is collaboration. These studies have shown the importance of collaboration. When you collaborate, you're able to use facilities that you don't have in your environment. You're able to broaden the science and things get better, and networking is also better. And you have wider diversity of exposures that you're going to look at. Third one is the importance of gene-environment interaction in disease process. And the best approach for doing this is a cross-cultural approach, which is what I'd adopted in all these studies. And the last point I want to make is dynamism in science, which is very important, that in Africa, we mustn't stop only with counting figures only. I think we must go to the next level of doing analytic studies and mechanistic studies, so we can then contribute significantly to risk factors and things that predispose to disease. And the only way we can do this is to remind ourselves of what Louis Pasteur said, that science knows no country because knowledge belongs to humanity, and it's the touch which illuminates the world. And I think is the last word that I'd like to leave with everybody, that you are in a low and middle income country doesn't mean you cannot do good science. In Africa, we are blessed with good environment. That's Kilimanjaro, and we have all the lovely animals and all that, and vegetation. And so we have lovely environment, and we can do a lot of work, and we should not restrict ourselves. And that's the essence of this presentation. And I wish to now end by thanking you all for your attention, but in particular, I'd like to thank Professor Benjamin Oshuntokunto, to whom I dedicate this talk. He was my mentor, was the one that set me on the path of dementia research. Unfortunately, he died shortly after we published the prevalence paper. I'd like to thank Hugh Hendry, the principal investigator of the research, Kathleen Hall, who took over from Hugh Hendry. This was Smith Gamble and all those that were involved, researchers in Indianapolis, Sue Gao, Fred Umversat, and others. And I also would like to thank my colleague. This is Oyegureje, a National Merit Award winner in science. This is Shegun Bayewu, that was myself at that time. I'd like to thank the elders in Idikon. We had this community of elders that ensured that there was community cooperation, and we had meetings with the committees that guided the research. And on this graph, I'd like to thank my field workers and the supervisors who participated in the study. I've concentrated mainly on the Ibadan study, but thereafter I got involved in the intervention and identification of dementia in elderly Africans, the IDEA study, which involved researchers in Kilimanjaro Christian Medical College, Tanzania, and Newcastle University. This is Richard Walker of Newcastle. This is Keith Gray, Catherine Dochin, and Sue Paddock. These are the individuals who assisted in doing that study. This is Declan Mushi from Tanzania, and this is my colleague in Ibadan, Akiadebi. I thank them all for their support. I'd like to thank NIH for providing the funds for the 20-year study. Then the Grand Challenge is Canada for the subsequent study from 2013 to 2019. And lastly, I'd like to thank the Lancet Commission on Dementia for the latest publication on risk factors for dementia, which is a good lesson for those of us in developing countries or low and middle income countries who are likely to face the challenges of dementia. Our publication shows that about 40% of the cases of dementia can be prevented if adequate attention is paid to some of these factors that could predispose to disease. And on this risk, we have hypertension, we have isolation, social isolation. So these are things, simple things that can work on, as well as the other things like alcohol, reduction of pollution, smoking, and increased physical activity. I thank you all for your attention, and I hope I've been able to impart some lesson to those who are early career researchers. Thank you very much.
Thank you so much, Professor. That was a very interesting and insightful talk. It was quite intriguing to hear how the research went over those 20 years. And also thank you for the inspiring words at the end of the talk. I think it's really important to motivate us Africans to start doing more, you know, as you said, analytical, mechanistic research back in the continent. So now we're going to move on to questions. So if anyone has questions, please type them into the Q&A. We already have a few that came in. So the first question, Abdullah asked, you showed data from Osantokun, 1994, showing that Nigerians had a lower percentage of people with amyloid compared to Australians. Did they use the same, a similar sample size for this study?
Yes, thank you, Abdullah. 111 Nigerians were compared with 99 Australians. So it was fairly similar. About 10 difference, more the Nigerians. And when they did the analysis, they ensured that they didn't look at the ages before they then did the analysis and then exposed the results later and then did all the frequency counts and the quantitation that I described. Thank you.
Thank you. I have a bit of a follow-up question from there. So in that graph that you showed from that same study, you showed that I think when individuals were in their mid-50s, there wasn't much of a difference in the amyloid between the Australians and the Nigerians. And then between 61 and 70, then you saw this decrease in the amount of amyloid in the Nigerian population. How do you explain that there was a decrease of amyloid from one age to another?
Yeah. Now, thank you very much for the question. Now, that was a curious finding, the amyloid story. The below 50, it was surprising that in the two cohorts, Australians and African American and the Yoruba, there was no amyloid. I think it's something that should be studied, that what triggers amyloid deposition after the age of 50 years, and has it remained the same? I think that would be the interesting bit. I can't answer that at the moment. Now, why there was a drop in the Yoruba, I think maybe it was a number, absolute number that was at that stage. But there was no other reason. But the thing that was interesting was the severity or the quantitation of amyloid in the brain specimens. None was found in the Yoruba, but it was quite 11% of the Australians had a lot of amyloid, severe amyloid deposition in their brain. Yeah, thank you.
Okay, thank you. So the next question from Ifeolua, they're asking, was there a comparison between the weight changes which impacted on dementia risk and the social economic factor?
We did not relate this to socioeconomic status because they were all resident in Idika. We just looked at the weight changes, not that they had any illness at that time. It was just doing the serial, each time we went, we took their weights and took the height and all that, and then computed the BMIs. And we then saw when we made the diagnosis of dementia that those that had cognitive problems had dropped their weight over time, which was not noticeable initially when we started. So it was a curious finding. Again, it's something that is open for research that I think others may want to take on. But I don't think there was any change in socioeconomic status in the community, in DCON, where we carried out all these studies at that time.
Okay, thank you for the answer. The next question, so in the GWAS study, were the variants identified in the Yoruba identified in any other population in the world? So I guess, were there any other comparative studies with other populations?
Yeah. Now, thank you very much. I like to answer that this was actually presented only this September for the first time at the Alzheimer Association International Conference that took place in the US. So it's fairly recent data. But I can tell you that the ATP binding cassette family had been described in African Americans before. That's not new. So, and a few studies have shown that clustering could be linked with dementia. So it's not unique to the Yoruba. It's just that this is new data and we haven't published it yet. I just thought I should complete the story when we said there may be other genes involved. That's why I thought I should add just that single slide. But I can provide more information on this if you email me with time. Thank you.
Okay, fantastic. So yeah, reach out to the professor if you want more insights on that. The next question from Joe is, can you explain more on the impact of social isolation on cognitive decline, or is it a hypothesis at this point?
No, it's not. It's not a hypothesis. It's actually been documented in literature that those who are socially engaged had better cognitive performance than those without. And in our study, in which we quantified, or we asked them about five involvements: social community activities, religious activities, social events, family prayers or family events, and I think five of them. Which one that said they participated in one, we gave a score of 1, and then we then looked at this and compared this with their cognitive performance, and we showed that those that had high cognitive scores or involvement scores performed better, and this is well documented in literature. So it's not unique to our own population. So it's well known, and part of the reason why we can explain that is that when you are socially engaged, you open up synapses, the brain reserve increases, and you get better. And so that is the reason, that's the explanation for that. Thank you. So it's not a hypothesis, it's real and it's well documented. Okay.
Thank you. I think that's a really interesting finding. And I think it goes to show the importance of social interaction and that we are social creatures and how we don't socialize, how it can have an impact on our.
And if I may say something again, Natasha, if you look at the Lancet publication that I just referred to, is one of those factors that we have listed as, if we do something about this, you can reduce the incidence of dementia by a certain percentage. So it's not just the Yoruba environment, it's universal. So it's something that we should recommend for elderly people, that they should be socially engaged. Thank you.
Yeah, absolutely. And I wanted to also ask, just generally because of the changing dynamics of the world and how, for example, diets are changing globally, people are starting to intake more fat-rich diets, even in Africa. Do you think that this is going to end up causing also somewhat of an increase in the prevalence of dementia in Africa in general, just because of the way we're starting to live differently?
I think so. I think that with the dietary changes, then vascular diseases, chronic cardiovascular morbidity will go up. And then you may have more risk of developing vascular dementias. And we know that most of the dementias have some vascular basis. So many of them are mixed dementias. So the more we engage in all these unhealthy diets, the more likely that we'll have problems. And then if we think of those diets resulting in cholesterol change. So even if you don't have E4 allele and your cholesterol level rises, then your chance of developing Alzheimer's disease will go higher. So it is true. So we must preach healthy diet for individuals, even in our own environment.
Okay, thank you. Another question from Biliam. Does environmental risk factors such as hypertension and dyslipidemia predispose individuals to AD or vascular dementia? I think you just touched on that now.
Environmental factors predispose hypertension, dyslipidemia, they predispose to Alzheimer's disease and vascular dementia. They predispose to both. And like I said, even those that we say have Alzheimer's disease have some evidence of vascular involvement in them. You have amyloid deposited in the vessels and all that. So it's not purely Alzheimer's disease only. So hypertension is a risk factor for Alzheimer's disease. Hypertension is a risk factor for stroke. Stroke will result in vascular dementia, but again, hypertension is a risk factor for both. So we should control blood pressure. And again, it's part of the Lancet 2020 report. Thank you.
Thank you. And then a question from Elaine. She asks, have you explored dementia biomarkers in other African populations? And if not, do you predict differences with the Nigerian population?
Thank you very much, Elaine. When we did, unfortunately, I didn't talk about our Tanzanian study. Part of what we wanted to do was to be able to do biomarkers and compare Nigerian Africans with East Africans and see whether we can bring out some environmental and genetic changes. Unfortunately, we didn't have funding for that, so we couldn't do that. So I cannot talk about these biomarkers in other environments, and other people have not done it. So it's a new area of research that people can go into, and I think interesting results will come out of that. So I think it's a promising area of research in other cultures.
Absolutely. Someone is asking for the link to the Lancet article. I'm not sure if maybe we can then share it after this with the participants of the talk.
Yes, I can do that. There is no problem. Like I said, it was a letter to the editor rather than a full manuscript. But it was quite interesting because it provided the basis for most of the things we did. And so I can share it. If I have your email address, I will do so.
Okay, great. So I wanted to, I think maybe if anyone else still has a question, please type it in. But I wanted to ask more question on career. What advice would you give to a young African who wants to pursue a career in research? So for those who are both on the continent right now who want to aspire to a career in research, but also some other Africans like me who have left and are doing maybe their PhDs elsewhere, but would want to come back in the future. Do you have any just advice or words of wisdom that we can use in navigating our journeys?
Well, I think it's nice to do research in Africa, despite the limitations and challenges, but you can go far because at least you can then look at the environmental factors. You can look at our limited technology and the things we can do with this, and you can then compare with what takes place in the high-income countries. So I would suggest comparative studies rather than studies just devoted to the African environment only, because then you'll be able to tease out differences and then think of ways of making a difference rather than just focusing here. It's nice to do all the training overseas and carry out some studies there. It would be nice to replicate them here as well and then see whether there are differences which can then be pushed further that will enable others to see whether there are areas to explore further in all these studies that we're doing.
I think another question. So we need to look into association between blood pressure variation and dementia. Is there any link between dementia and malaria fever? Are you aware of?
I'm not aware of any link between malaria, fever, and dementia at the moment. No, I'm not. But it would be nice for some people to take it up and look into it. But malaria is so pervasive that if there was going to be anything, then by now virtually everybody. So, but there may be other things that we can look at, maybe some biomarkers and see whether they play any role in the pathogenesis of the dementia syndrome.
Okay. Okay, there don't seem to be any more questions. So I think with that, I'll close the session. So I would like to thank you again, Professor Adesola, for this amazing talk, for answering all these questions and for just inspiring us and giving us a lot to think about, especially thinking about getting back to doing some research back in Africa and doing these comparative studies so we can better understand also what's going on with dementia within our own populations. So yeah, thank you so much. And thank you for everyone who attended today. We'll be back again next month, the last Tuesday of the month, with another talk. And I wish everybody a pleasant morning, afternoon, evening, depending where you are.
Thank you very much, Natasha, and thank you, IPGC, for allowing me to do this. I'm very grateful. Thank you. And thank you all for listening and participating, and for all the various questions. Thank you so much.